CellPort is the world's first all-in-one LIMS built for cell culture, cell banking, lab operations and bioprocessing. In this video, get a brief demo of CellPort. We're looking at the CellPort calendar, which is how most users manage their daily cell-based operational activities. The CellPort calendar shows a day-by-day list of all the activities that you have completed, all your activities in progress, and all your scheduled events that haven't yet been started. In the CellPort calendar, you can see information on the passage numbers of your cells that need to be fed or passaged, as well as equipment that is due for calibration. You can filter the CellPort calendar to view activities for your whole team, giving you an organization-wide view of your cell-based operations. You can also view activities that have yet to be assigned. Let's assign one of these unassigned events to ourselves. Once assigned, the protocol that is associated with the newly assigned event can be launched directly from the CellPort calendar. Let's launch the PC12 cell passaging protocol for Flask of Cells. Protocols in CellPort are comprised of steps from a protocol template that has been created by one of your on-site administrators. Your admin has granular control over each step, including which fields are required, which fields are optional, whether or not steps must be electronically signed and witnessed, and whether or not one step must be completed before the next step is started. We see that when we launch the protocol, it receives a date and timestamp along with the name of the person who launched the protocol, part of the traceability and transparency that are indelible aspects of CellPort. The first step in this protocol is a simple instruction, specifying that the user must clean the biological safety cabinet according to the approved standard operating procedure. Completing this step results in a date, time and user stamp for this step's completion and allows us to go on to the next step. If a different user were to log in and complete subsequent steps, their username would appear for all steps completed by that user, again ensuring indelible traceability and transparency in CellPort protocols. The next step is an inventory check-in step, in this case, scanning the CellPort barcodes of all the cells to be passaged, the growth media, and a pipette filler to check them into the cleaned BSC, whose barcode is also scanned, to confirm the specific BSC being used. Note that the cells being checked in are at passage number 6. Location history, whether automatically documented via a protocol inventory check-in step or manually entered, is tracked and viewable for all materials in CellPort. The next step is a centrifugation step, which instructs the user to centrifuge the cells. The admin who authored the template for this protocol has chosen to pre-populate the actual RPM and time values with the target values defined in the protocol template to make completing the step faster. You can always make adjustments to the actual values before you save the step if you deviate from target and want to capture the actual results. The next step instructs the user to aspirate off the spent media, then to resuspend each pellet of cells in fresh media. We record the specific lot of material that was added to the cells in this step, as well as the specific pipette filler that was used. Again, part of the indelible traceability and transparency in CellPort protocols. Note that we didn't have to rescan the barcodes for the media or the pipette in this step. The admin who authored the template for this protocol used an inheritance option to transfer information seamlessly from the second step to the fourth step, a powerful no-code configuration technology available for your CellPort admins to use when required. The next step is a measurement step, capturing the cell density of the resuspended parent cells that are being passaged. We need to enter 2 replicate values here, which are averaged and saved as a measurement for the parent cells. The average cell density measurement value from the previous step is inherited in the cell passaging step, along with the parent cells and media. Here, The admin who authored the template for this protocol specified passaging suspension cells using a seeding method of cell density in the child flask specified in units of cells per millimeter. Admins can set the cell passaging step in a protocol template to passage either adherent or suspension cells using a target of either cell density, which for adherent cells is expressed in cells per centimeter squared, or number of cells. Once the actual values for the volume of cells from the parent flask and the volume of fresh media added to the child flask have been entered, clicking the Create Lot button commits the data to the system, creating a new lot of child cells with a new material ID, new lot number, and a passage number incremented by 1 to 7. The next step is an inventory check-in step to record checking the child flask into an incubator. Scanning the barcode of the incubator allows us to choose the shelf to which we added the new flask of child cells. Next, we record checking the media back into a refrigerator. After scanning the barcode of the refrigerator, we can choose the shelf to which we returned the PC12 media. The remaining steps of the protocol are for recording the passage of the parent cells as well as to schedule 2 feedings and a subsequent passaging of the child cells. The current step records the passaging of the parent cells as an event that will be associated with the parent cell's material record as a part of its permanent history in CellPort. The next 3 steps allow the user to schedule a first feeding of the cells in 2 to 3 days, a second feeding 2 to 3 days after the first feeding, and the next passaging in 1 week. Setting the overall protocol status to completed allows it to be finished. Now, let's look at the effortless self-documentation features that add valuable traceability and transparency through the use of CellPort protocols. Looking at the child flask material that was just created in this protocol, we see the scheduled events for the 2 feedings and the next passaging, including the protocol templates that will be launched when these events come due, as well as the protocols that were run to schedule these events. Looking at the media that was used in this protocol, we see changes in location history and quantity history for this material, including the users, dates, times, and protocols involved. CellPort protocols, aided by quick and accurate inventory barcode scanning, makes it effortless to maintain detailed historical records of who did what, when, where, and with what materials and equipment. Returning to the CellPort calendar, we can see that the protocol that we just completed is marked as done, and the 2 feedings and the next passaging are queued up and ready to go. Your administrators will always be able to keep your protocols up to date, with the ability to create new versions of protocols with strict version control, or they can clone existing protocol templates to quickly adapt them to new cell lines or experiments. Also, if you have expert users who want a faster user experience for entering data, your admins can choose to create protocol templates in expert mode, which streamlines the process for quick, research-based protocols run by your most skilled team members. CellPort was created with cells in mind, including the ability to set cell line-specific passaging limits with cell line-specific thresholds to alert you to cell flasks that may have been passaged too many times. The intuitive user interface in CellPort allows you to find the information that you need with 1 or 2 mouse clicks. Software for powering breakthroughs, built by our scientists for your scientists. That's CellPort.